Hello everyone, welcome to our Avantor webinar. We will start the webinar in 1 or 2 minutes as there are still people joining the call or the webinar. Hello again from my side. Welcome to our today's webinar. My name is Martina Witzenbacher and I'm here at Avantor site, the portfolio manager for chromatography consumables. And together with my colleague Didier Sember, we are the host of the today webinar. Today we will be hearing in our webinar about hydrophobic interaction chromatography, a versatile method for ADC and protein analysis. The presentation will explain how HIC works, presenting some applications example and providing tips and tricks for the method development. Our speaker today is Andrea Krumm from Tosoh Bioscience. Before I hand over to our speaker, some couples of housekeeping information from my side. We will send you the recording after the webinar, so you will get an email afterwards with the recording for you to listen again when you want to do it. If you have questions, and I'm nearly sure there will be many questions during the presentation, you can enter them anytime by typing them into the question tab. You see this in the menu on the right side of your screen. Question which needs to have further works, further investigation or research, we will handle them to you after the webinar. We will have time after the session to ask some questions direct to the speaker. If you have some additional questions after the webinar, please do contact us at webinar@avantosciences.com. You will get this address from the mail after the webinar anyway. So far, so good. Coming to our today's speaker. Our speaker today is Andrea Krumm from Tosoh. Andrea studied biotechnology before gaining a PhD in cell and cancer biology. She joined Tosoh Bioscience in 2020 as a product manager for analytical columns. As such, she is responsible for gathering customer requirements, application areas, and addressing this with new and exciting products of the analytical column line. Today, it's a great pleasure for us to welcome her to our webinar. So Andrea, the stage is now yours, and I wish you all a great and interesting webinar.
Thank you, Martina. Thanks a lot for the very nice introduction. And of course, thank you to everyone who joins us today. And also, I would like to thank Avantor for actually giving me the opportunity to talk to you today. And the topic will be hydrophobic interaction chromatography. And it is actually a topic that can do some promotion because it's a very versatile method, as we will see in the next 30-35 minutes. Just a small reminder of what we will hear. So I will give you an overview on how hydrophobic interaction chromatography actually works. Then some examples of what it can be used for, which is quite a variety of applications that we will see there. Then we will rather look into the how it works and look into which parameters can we change in order to optimize our method we use for hydrophobic interaction chromatography. And I will end with a very short summary. First of all, how does hydrophobic interaction chromatography actually separate molecules by hydrophobicity? Because this is what is done. HIC separates molecules by hydrophobicity, meaning the molecules that are less hydrophobic will elute earlier, and the ones that are more hydrophobic, they will elute later. And in order to get this separation, we will use a column with a stationary phase that has a unpolar or hydrophobic surface. And we apply our sample in a very high salt concentration. And this high salt concentration leads to the fact that our molecules are hardly solvated, and all of the hydrophobic parts that we will find on the surface of our molecules, on our proteins, for instance, are accessible. The same is true for the hydrophobic surface on our stationary phase. It is accessible, so both hydrophobic parts of the protein and the stationary phase can interact, and we have binding of our molecules. Now, in order to elute our hydrophobic molecules again from the column, we simply reduce the salt concentration. That means we get solvation of the stationary phase and of the hydrophobic parts of our molecules, and thus we don't have the interaction anymore between those hydrophobic parts, and we will get elution. Now, a very similar but better visualized, I would say, illustration of what happens for the binding of our molecules, in this case proteins, onto the stationary phase is shown here. Here I added salt, so one of the salts which is often used in order to use it for HIC is ammonium sulfate. And so what we will have is like all of our water molecules, so these little guys here, they will be kept busy by all of the ions that we have because of the high salt concentration. So there's basically nothing left, or everything is freed in order to interact with salt, and we don't have anything attached or close to our hydrophobic parts anymore, and we do have the binding. In contrast, this is what I also told you in the slide before. If there is no salt anymore, we have a lot of water molecules that can solvate our protein and the hydrophobic parts of our proteins, so we don't have this accessibility anymore, and we do get elution of the molecule. Now, one question we always get, or we very often get in particular from researchers that worked a lot with small molecules or reversed phase, is why do I actually need hydrophobic interaction chromatography? Because I know a method, reversed phase, that also separates my molecules by hydrophobicity, but I have more experience with it. There are more papers out, there's just more knowledge on how to get a good RP method, in contrast to hydrophobic interaction chromatography, where there are many variables that I need to take care of during my method development, and there's not so much expertise on HIC. But the important point is that if we are working with proteins, they will denature if we use reversed phase due to the use of organic solvents. So what we denature is the alpha helices or beta sheets, and we do that irreversibly. And this is something that we can prevent using just a salt buffer. This is why HIC is a very popular method for protein purification. It can also be very nicely used, as we will see, for the analysis. There's just one, I would say, limitation, reversed phase, due to the fact that there's often no or just very low salt concentrations used. It's very nicely compatible with mass spectrometry, which, due to the high salt that we use for HIC, this method cannot be combined with mass spectrometry as easily. Now we come to the molecules that we actually want to look at, proteins. Why can we separate proteins by hydrophobic interaction chromatography? Why are they retained? So what we will find if we look onto the surface of proteins is that there are parts of proteins that are more hydrophilic, shown here in blue, and there are parts that are more hydrophobic, which is due to the different amino acid side chains that are on the surface of these proteins, meaning these patches can actually interact with the stationary phase of our HIC column. So what we find is different hydrophobicities for different proteins. But what we will also find is that modifications on proteins also has an impact on the overall hydrophobicity of the molecules. And these can actually be very tiny modifications, as we will see, that lead to such a shift in hydrophobicity that is detectable by HIC separations. One of them is, for instance, deamidation. So this is shown here from asparagine to aspartic acid. What we see is that this amide group turns into a carboxyl group. So we have a hydroxy here, which is less hydrophobic, more hydrophilic, as opposed to this. So if we have deamidation, we do get a decrease in hydrophobicity. Same is true if we look at methionine oxidation, which means that sulfur and methionine is either once or twice oxidated. Again, we add oxygen, which leads to a decrease in hydrophobicity, or the other way around, increase in hydrophilicity. Now, what we also find is that the size of a molecule has an impact on how hydrophobic it is. For instance, fragments or smaller molecules are less hydrophobic as larger molecules. This is something we find, for instance, for fragments of antibodies or dimers of antibodies, but you can also see it generally if you work with very small proteins or even peptides. These are very low in hydrophobicity, whereas large molecules are more hydrophobic. Last but not least, and this is common sense, I would assume, if we modify our molecule, in this case an antibody, with a chemotherapeutic drug, for instance, which is more hydrophobic, we also add hydrophobicity to the molecule and increase the overall hydrophobicity of our molecule. And with that, I am already in one of the first applications, because this is like the main application of hydrophobic interaction chromatography, and this is the analysis of antibody-drug conjugates. So antibody-drug conjugates, you very likely know, it's basically an antibody that can specifically bind a target molecule, a target antigen, for instance, a protein on a tumor cell, and this is attached to a chemotherapeutic drug. So it basically delivers the chemotherapeutic directly into the tumor. This hydrophobic makes the molecule much more hydrophobic, and of course it's important to know how many of these drugs do I have attached to my antibody, because this will be the actual drug that elucidates its mode of action. Now, how many drugs on average there are on an antibody is called the drug-antibody ratio, and this is something you can determine by hydrophobic interaction chromatography. So as the more drugs there are on the antibody, the molecules will elute. Later, you can very nicely separate them. If you quantify the peak areas underneath these, you can calculate the average drug antibody ratio using hydrophobic interaction chromatography. Now, another thing we were looking at was deamidations. And here is an example of how that could look like if identified by Higg. So what we see here is a fragmented or digested antibody into the Fc and both Fab arms. And what we are looking at in these three chromatograms is the cation exchange fractions of the whole antibody, basically. So cation exchange is an orthogonal method to look at deamidation. So what we wanted to try with that is to get fractions that are enriched in the deamidated versions of our, in this case, Fab arms. What you can see in the middle, this red line, the red chromatogram, is the main product that doesn't have any modifications. And we have the green line, which basically shows two peaks, because we do have an aspartate in this Fab arm that is modified to isoaspartate. So basically from this to this, and this has an impact on the hydrophobicity, meaning we do see separation over here. Very similarly, we have in a third cation exchange fraction an enrichment in the modification from asparagine to aspartate. So from this molecule to this molecule, also having a change in hydrophobicity. This is why we see two peaks over here. So to conclude on this slide, we can separate Fab arms that have deamidations versus the original asparagine, for instance. Now with that, another very small modification. Here it's a little easier to detect it actually, or to induce the oxidation, because what we did here is we can digested a full antibody into the Fc part and the Fab arms, and those digest we treated with a oxidizing agent. So from 0%, so no oxidizing agent, up to 0.1% of the oxidizing agent. What we will see here in the Fc peak, actually the Fc part is the part where we have the methionine. We do see that we do get induction of the oxidized parts of methionine. So we have then the main product without any oxidation. Then if we have a lot of oxidizing agent in there, we do see that a one-time oxidized molecule comes up and a 2 times oxidized molecule. So this was now looking at very small changes in or very small modifications in the molecule. Now let me show you some applications where we look at like bigger changes in our molecules. For instance, to look at the success of Fab exchange in bispecifics. So bispecifics, as you probably know, can be composed out of basically 2 parts of 2 different antibodies. So what we see here is we have a monospecific antibody, another monospecific antibody that targets another molecule, and then by Fab arm exchange, we basically exchange those 2 arms and we get a bispecific antibody that targets both antigens. Now, as the monospecific antibodies, so the parental antibodies, differ in hydrophobicity, we can see in hydrophobic interaction chromatography whether or not the exchange was successful. So what we see here in blue is our one antibody. What we see here in yellow is a second antibody, and basically the mixed bispecific we see in black. And we do see it was quite successful, as we only have a little of the monospecific ones left over here. With that, I would like to come to last application, which looks into the hydrophobicity between different molecules. Why is this important? Because when you're first screening your molecules or your drug candidates, it is important also to have a look on the hydrophobicity. Why? Because higher hydrophobicity leads to a higher aggregation propensity. It has an impact on the solubility of your molecule, and it also impacts on the drug clearance. So if the molecule is too hydrophobic, it is very likely it won't succeed in clinical studies. So, and this is something, just because of the overall hydrophobicity, you can already determine at very early stages. And this is something which is shown here. Different molecules were analyzed as to their overall hydrophobicity, with some that are very hydrophobic, some that are less hydrophobic. So what we have here is the different molecules, and here's the retention time. So the ones that have a low retention time are less hydrophobic as the ones with higher, of course, and some didn't even come off the color. So this is a very nice method to initially already decide, is a molecule worth to continue with or not? And now that you know what you can do with hydrophobic interaction chromatography, I hope that at least one of the applications is something that's relevant to you as well. I would like to go into how can we actually do that and look into how to develop a method. So first of all, we need to decide on a column that we use. How do we do that? So as for the columns as well, we have a selection of different functional groups. So what you will often find for proteins or antibodies is actually brutal functionalization, but there are also others out there. And it's important here to say that generally for molecules that are more hydrophobic, you need a column that is less hydrophobic. And the other way around, if you have a molecule that is hardly hydrophobic, you will need a column that is very hydrophobic. And of course, you get an impression of how hydrophobic the method is by just the functional group. But I would like to point out here that it's not only the functional group which has an impact on how hydrophobic overall our column will be, because it also depends on the ligand density and on the base matrix. So phenyl and ethyl, for instance, these are functionalities that Tosoh, for instance, has for hydrophobic interaction chromatography columns. But the ethyl one, for instance, is more hydrophobic as compared to the butyl one, just because there are more ligands on the surface. It has a higher ligand density as compared to the butyl one. So what do I want to say with that? It's not only the functional group, it's more that plays into how hydrophobic a column is, and it's very hard to compare hydrophobicities just based on the functional group. Just like for other columns, you will find different base materials, so like the particle that we find in our HPLC column, basically. So we have polymeric base materials, for instance, polymethacrylate, silica-based ones, non-porous and porous. The most popular ones are, again, the polymeric columns, because they are stable, they can be used at higher pH, for instance, which is not the case for silica-based ones. And we've also find a preference typically for non-porous columns, just because they have a very low mass transfer resistance, which is why they show very sharp peaks, at least for hydrophobic interaction chromatography, very sharp peaks, and fast analysis, whereas porous columns are rather used for semi-preparative applications. Now, looking at what is the most popular characteristics, it's butyl columns, a polymeric with non-porous material. And I would like to just take a single slide, so not more than one slide, to do some advertisement for a new hydrophobic interaction chromatography column that will be available as of next year. So it's called TSK Gel HIC ADC Butyl, and as the name already says, it was really developed for this ADC analysis of the drug antibody ratio. As I said, it's a non-porous one. It has, as the name says, a butyl functionalization. And what we saw is actually that we will achieve a very high resolution on that specific column. Compared it to other columns that are on the market, silica-based ones, porous ones, non-porous ones, with polymeric particles, so everything which is out there. And what we found is actually that we get a surprisingly good separation performance. For instance, if we look into the R4 peak, we do see this small shoulder that is not visible in any of the other columns. Same is true for R6, where we have double peak basically, and also this can hardly be seen on any of the other colors. On top of that, we also looked into how reproducible those analyses are. Here we injected an antibody 500 times, and we do see the very same peak shape over 500 cycles. And last but not least, due to the fact that this column can stand high pressures and high flow weights, we can really accelerate the analysis, for instance, of the drug antibody ratio. This is what has been done here on the 3.5 centimeter column. And we can analyze the DAR in 7 minutes, whereas typically it takes like 20 minutes to analyze the drug antibody ratio. As I said already, available from next year on. And with that, I would like to switch again from the advertising mode into the method development mode. One of the most important things we may want to look at for hydrophobic interaction chromatography is the question, which salt we use. I said it very initially already, what we typically use is ammonium sulfate, but we can play around with it. So what we will find is rather cosmotropic salts, and cosmotropic means we have increasing precipitation and increasing interaction with the stationary phase. So if you find that a molecule doesn't bind to your stationary phase or is not retained, you may want to try something that is rather in this region of the Hofmeister series. On the other hand, if we use rather chaotropic salts, this decreases the precipitation and the interaction with the stationary phase, which is, again, if you have something that basically sticks to the column, rather think of choosing something in this region. Now, here is one example of how different salts impact on the separation. So what we analyzed here was, in both cases, a map, and we also included a stressed map in order to expect, or where we knew there would be aggregates in there. And as I said initially, aggregates are more hydrophobic, fragments are less hydrophobic. So we should see a separation on there. But using, in this case, ammonium sulfate, we hardly see anything, neither in the mAb nor in the stressed mAb. This changes if we go to sodium chloride, because there we can see the separation of our fragments and of our aggregated protein. What you can also see is that we do have a different concentration. This is due to the fact that this is more chaotropic, this is more chromatographic, so we need less of this, that is has a higher precipitation it's more prone to precipitate, put it that way, than the sodium chloride. Now, another thing, apart from the fact which salt works and which does not, the concentration also plays a big role. So there's some rule of thumbs that help us to find the right concentration. So in general, we should always use the lowest salt concentration that is possible. Secondly, smaller proteins, as they are typically less hydrophobic, require a higher salt concentration as compared to larger proteins. So if you think of a 20 kilodalton protein, you may need 3 molar ammonium sulfate, whereas a 150 kilodalton mAb only requires 1 molar. Also, and this is also becoming clear if we look into the Hofmeister series with the chromatographic and chaotropic salts, chromatographic salts require lower salt concentrations as compared to chaotropic salts that require higher concentrations, for instance, sodium chloride. This was also something we have seen on the slide before. Also, what we see here for a separation is that higher the salt concentration gets, the later the elution of our molecule will be, though there is, of course, a critical concentration where our molecule doesn't bind at all, for instance here at 0.7 molar. Now, another thing we can change or we can play around with for developing our hydrophobic interaction chromatography method is the pH. It's typically not something you think of first when doing the first run, I would say, in HIG, but it can be very helpful. So typically what we tend to do is to use a pH which is close to the isoelectric point of our molecule, just because at that point there should be no net charge, which is why we don't separate by introduced hydrophobicity changes by charge, because charges do decrease the hydrophobicity of the molecule. But we really separate by hydrophobic interaction chromatography because the net charge is 0. However, if we think of a solution with 1, 2, 3, 4, 5 different molecules over here, that all elute at the very same time at a given pH, don't have any separation on here. If you think of like all of these 4 molecules are eluting between, let's say, 12 and 15 minutes, we hardly will see any separation. However, if we further decrease the pH, we will see that all of them are eluting at a different time, meaning we do get separation of this mix of molecules. And with this, I would just come to some general tips and tricks for hydrophobic interaction chromatography, one of which is it's important to think of which molarity is my sample in before injecting. Because you can imagine you do need a specific salt concentration in order for the molecule to bind. And if you just have it in pure water, for instance, it will not bind at all. And this is what we see here. So if we have our sample without any salt, a lot of the sample will already elute at the very beginning and will not be retained. The closer you get with your molarity in the sample buffer to the molarity in the buffer, the more will also be retained. A question we often get is, how do I design the gradient? And just as a rule of thumb, or as a way to start, a linear gradient from 100% of your salt buffer down to 0% of salt, basically water, in 10 column volumes, linearly down. One thing I wasn't referring to now is that you cannot only use different salts, but you can also mix different salts in order to get another selectivity. Of course, this will be a little try and error because it's very hard to predict what mixing of different salts may have as an impact, but it's sometimes the key to a successful Higg method. Now, due to the fact that we have a lot of salt in our buffer, especially in the A buffer, so the buffer for binding, basically, it is recommended to filter the buffer before you attach it to the HPLC system, just to remove any salt crystals. Also, always when working with high salt concentrations, it does make sense to, before running it for the first time, mix your sample once with the buffer for binding, or buffer A, just because it might happen that your protein precipitates in the high salt. And this should not happen, of course, or you don't want it to happen in your HPLC instrument, but outside of it. If something like that happens, again, choosing another salt, reducing the salt concentration could be something that might help you. Last but not least, this is also something we learned. If we use small particles and non-porous materials, we can increase the separation performance due to the low mass transfer resistance we see in non-porous materials. And with that, I am already through the introduction to hydrophobic interaction chromatography, and I would like to get back to the title actually. What is hydrophobic interaction chromatography? It's a separation mode that separates by surface hydrophobicity just using a salt grade. Why should we use it? Because it can determine a variety, I hope I could make it clear, a huge variety of protein modifications. And how we do that? We develop our method that is mainly focusing on the salt that we use in there, on the concentration, and of course some other things as the pH, as I mentioned already. And with that, thank you a lot for your attention. Would also like to point out, if you have any challenges with your protein analysis, feel free to reach out either to me or of course to Avantor, and I am happy to take any questions.
Thank you very much, Andrea. Thank you very much. It was really complete, really interesting, and I've learned a lot. So yes, we have received some questions, and if you want, I will ask you. And before asking the questions, I would like to introduce myself. So I'm Didier Sampere, working in a global marketing organization to push chemicals and all the ranges for chromatography. Okay, so now I would like to ask you some question, Andrea. So the first one, is there a typical salt concentration for mAb analysis?
Yes, I would say so. So, I mean, the very typical salt which is used is ammonium sulfate, and there we typically end up with like 1 molar or 1 molar to 1.5 molar of ammonium sulfate. Of course, if you change the salt type, for instance, to sodium chloride, this is something we saw, then it increases. So for sodium chloride, it would probably be something like 3 molar you needed to add.
Okay, thank you. Another question from Germany. Can organic solvents be added for ink separations?
Yes, they can be added with some caution, because you can imagine if I have a very high salt concentrated buffer together with an organic solvent, it may be prone to aggregation, so to precipitation. So you really need to check it up front again in order to see if you have any precipitation. But in general, it works, and one popular way to use it is to add it, for instance, into the elution buffer, so to buffer B, so that you have an accelerated elution from the column, or that you have better elution from the column.
Okay, another question. Can IC be combined with MS?
I would say no, due to the fact that we work with a very high salt concentration, which is not beneficial for the ionization of your molecules, neither for your ionization source. Though I think there are 1 or 2 papers out there that showed you can connect both, that work with with lower salt concentrations and instead used some pH shift. But yeah, it wasn't the most forward way of working, I would say.
Okay, and the last one. Was the new butyl column tested with molecules other than ADC?
Yes, we did test it. We did test it with other proteins, and actually it worked very well. So what we used it for was monoclonal antibodies without the attachment of a drug, or just simple protein standards, for instance. And there we also saw like that superior separation performance, resolution of the column, just like for the ADCs. But yeah, it's all been protein-based molecules up to now.
Okay, thank you, Andrea. So thank you very much again, and thank you also to Martina. It was a great introduction. And now I would like to give you additional information concerning the brochures. So you have 2 brochures loaded in the process, so you can be free to download the brochures. They're very, very interesting and linked to this topic. And after, you have also the possibility to go in the literature part in our website and to find the different brochures created by Tosoh. You can go in the chromatography part and after write Tosoh, and you will find all the different brochures they have for different applications. Okay. Next point is that tomorrow you will receive the recording, so feel free to share with your team. I'm pretty sure that many people in your lab will be really interested by this presentation. In some minutes you will receive a survey, so feel free to give us your feedback. It's really important for us to know your opinion, because yeah, with that we can improve the next webinars. You can also write some suggestions for some future webinars. So yeah, feel free to fill this survey. It's still important for us. And the last point is that in 1 week you will receive a follow-up mail with many additional information on brochures, on link to the website, on product page, etc., etc. So yeah, you will discover this mail very, very soon in 1 week. Okay. So I think that we are finished in advance. Andrea, do you want to add something to your presentation?
No, I hope everything was clear, and yeah, thanks again for letting me talk today to everyone. Yeah.
No, thank you very much, and again, thank you to Martina and also Florence for the organization of this webinar. So thank you very much. I'm sure that our customers will be more comfortable on this application. They have discovered many tips and tricks, and and now I would like to close the session. And before that, I would like to wish you a great evening. Thank you very much, and not see you soon, but talk to you soon for next or future webinars. Thank you. Bye-bye.
Thanks a lot.
Bye.
Hi, bye, Didier.